I'm standing inside a very special plant growth chamber. Around me are very special mustard or brassica plants. About 2 decades ago, India embraced genetically modified BtCotton, and since then, BtCotton has been a success story. Now, there are efforts at making newer and better plant varieties using modern genetic engineering techniques like gene editing and using them for increasing the crop production. India tried to introduce genetically engineered brinjal and genetically engineered mustard. This episode is all about understanding genetic modification, genetic engineering, the 21st century way of increasing the output in agriculture. Should India embrace genetic modification in crops or allow genetic engineering in food crops? This is one important question which the country is grappling with. We have with us today Professor Deepak Pental. Namaskar, sir.
Namaskar.
Welcome to the show.
Namaskar.
Professor Pental is a plant geneticist and somebody who has worked on genetic modification of an important oilseed crop, mustard, a Gandhian by values, and also you have a species of frog named after you.
Yeah, that's Dr. Biju's job.
Professor Pental, India is a very large country with a large population, 1.3 billion plus. Is our food production keeping pace with our population?
Yes, certainly cereal production is keeping pace with the population. In fact, we have surplus of rice and wheat. But when we go to other crops, oil seeds, even coarse grains, legumes, fruit, vegetables, I think we need much more than what we are producing today.
But what about in the future? Because India is touted to be becoming one of the world's largest populated countries.
You see, the research intensification in rice and wheat is very high. Rice, wheat, maize, these crops are globally being worked upon. But when it comes to our own crops like mustard, and also I would say soybean, it's an introduced crop, but it's a very important crop of central India. So on these crops, our R&D work is not of the intensity that we require. And I think recently DBT has given a lot of germplasm projects, but that will take 10 to 15 years to come to fruition. So we need research intensification in these crops. We need to get germplasm from outside the country also. We need much more effort on these crops.
But what about oil seeds, which is your speciality? India is not self-sufficient in oil seeds.
Well, it's very unfortunate that we import around 70,000 crore worth of edible oils for the last 4-5 years. Our farmers should be earning that money, and the yields of the 3 major oilseed crops, groundnut, soybean and mustard, are almost stuck. Mustard has shown little bit improvement with little bit irrigation expanding in Rajasthan, but these crops should be yielding 2 to 3 times more than what they are yielding. And that is when they will become a good alternative crop to rice and wheat.
Professor Pental, is genetic modification and genetic engineering a solution for low input, high output agriculture?
It is one of the solutions. If you can have the best of the technologies, and if your management is poor, you'll make a mess of it. But Pallav, we must look at it this way. Every crop has got 5 to 6 major problems. For example, mustard has white rust, mustard has alternaria blight, mustard has stem rot, which is a newly emerged disease. Then we have aphids problem. You take rice, blast is a problem, sheath blight is a problem. So every crop, wheat rust diseases are a problem. To tackle these diseases and pests, we need solutions. And the solution is either agrochemicals or genetic means. And in genetic means, if you want to do it genetically, which is more benign, genetic engineering is one of the key areas in which India should be excelling. In fact, we should not be also ran, we should have excelled in it.
India embraced genetic engineering and genetically modified crops through genetic modification of cotton, which has been released in the farmer's field. Tell me a little bit about the Bt Cotton affair, and we will talk also about Bt Brinjal and mustard as we go along.
You see, the Bt Cotton was only about one particular insect, Helicoverpa. It was not about jessets, it was not about whitefly. So it is controlling even today, since its release, the most important pest of cotton, Helicoverpa. Just imagine how much pesticide we would have had to spray, and it is helping other crops also. Don't forget, our chickpea and pigeon pea are highly susceptible to Helicoverpa, and because the Helicoverpa population is being controlled on cotton, it is having a salutary effect on the cultivation of our legume crops also. This is not easily mentioned fact.
Correct.
This is very important. The ecosystem services that this Bt Cotton is providing to other crops also, which are highly susceptible to Helicoverpa.
Now, have farmers embraced Bt Cotton?
Farmers accepted it when it was not even released. In fact, they forced the government to release it. And if you want them to leave it now, you will have to spray more pesticides to control Helicoverpa.
We also had this, I would say, an foray or an encounter with Bt Brinjal. And brinjal or eggplant is eaten by a Bagla.
Brinjal is a much smaller crop, but the amount of pesticides farmer is putting into brinjal is horrendous. And brinjal cannot be compared to cotton, which is grown in 10 to 11 million hectares. But if brinjal protection would have been allowed, people would have been ingesting less of pesticides, which are being sprayed to protect the crop right now. Pests are going to be even a bigger issue with the global warming, and all kind of papers make the right predictions that pest incidence is going to increase only. So pests you can only deal with either with chemicals or with genetic means.
Sure.
That is the country's choice.
But this moratorium on Bt Brinjal, was it a good idea or was it a bad idea?
See, my point of view is that GAC should function,
which is the Genetic Engineering Appraisal
Committee in the Ministry of Environment. It should base its decisions only on scientific merit, and then the governments can decide whether they want to release something or not.
Now, your key area of work is genetic modification of a very important crop for India, oilseed, which is mustard. And how long have you been working on mustard?
Almost 30 years. But genetic modification can be done by conventional breeding. That's also genetic modification.
Correct.
In fact, I prefer to use the word where recombinant DNA comes in as genetic engineering.
Sure.
Because these are new technologies.
Sure.
Classical breeding is also genetic modification. So the use of word GMO is very unfortunate. in my view, very unfortunate. We should differentiate between conventional breeding methodologies, which is also genetic modification, and genetic modification through GE technologies, which are based on recombinant DNA revolution, of which we are hearing now all kind of vaccines which are being used for control of COVID disease. They are all based on recombinant DNA technologies.
Sure. People have embraced mRNA vaccines, DNA vaccines.
That is because it is us. We want to save ourselves. And that is a dilemma, Pallav. If you look at the global population, it was 1.6 billion in 1900. And India alone in 2040 is going to be 1.6 billion. So look at the population explosion. And how are you going to feed all these people? So you need all kind of technologies. You assure of safety, but rejecting a technology, I think, is foolish, and the future generations will not forgive these people who are opposing these technologies, because there will be massive problems of feeding people.
Having learnt about the big picture on genetic engineering and food crops across the world, let us now take a look at the actual development work happening on genetically engineered mustard in Professor Pental's lab and the growth chambers. Let's go. I'm inside the tissue culture laboratory where Professor Pental has been working on creating the genetically engineered mustard crop. Professor Pental, what exactly is your research work on genetically engineered mustard?
Well, the project is on many aspects of mustard. One is to create new variation. As you know, that is the essence of plant breeding. A lot of plants you are seeing here are the double haploids, which are recombinants between Indian types of mustard and East European type of mustard, Indian mustard and Chinese type of mustard. Idea is to increase the genetic variability and then select from it better plants. So that's one point. The other, of course, is our work on the Barney's bar star system as a pollination control mechanism for hybrid seed production. That is long over. The technology has been developed, it has been looked at for biosafety aspect. It was even GAC asked for its release, but then somehow people backtracked, and then one more test and another test and another test. So genetic engineering right now, we have made some transgenics for resistance to glyphosate, which could be very useful for conservation tillage. If you want to sow after rice mustard, you can sow it in the rice fallows and spray the herbicide to control the weeds, and also a very threatening root parasite, orobanche, which can be controlled by glyphosate. So as I was telling you earlier also, there are number of objectives in each crop, and some of those objectives can be met with conventional breeding, and the other objectives you very badly require genetic engineering and gene editing technologies, gene editing being the new technology on which all the kind of excitement is going on all around the world.
Now, can you explain very briefly what is the difference between genetic engineering and gene editing?
Genetic engineering is putting a foreign gene into a plant. It could be a bacterial gene, as was the case of BtCotton. For glyphosate resistance also, it's a bacterial gene. But more and more people are putting plant gene into plants, because earlier plant genomes were not so well explored. Now every crop which is grown around the world, including its wild germplasm, have been sequenced right now. So we are sitting on a huge pile of data, and how to make use of it through genetic engineering, through gene editing. Now, in case of gene editing, no foreign DNA remains in the plant. It's a process which you can call as much more efficient mutagenesis. Earlier, we were irradiating the plant. It may hit the gene you want to hit or not, and it may create some ancillary mutations in the plant. But now you can say this is the gene, I want to mutate it to make it like the resistance conferring gene in the relative of the in the wild relative of the plant. If you want to bring the gene from the wild relative, it will take you about 12 to 15 years.
12 to 15 years.
Absolutely. People don't understand how tough plant breeding is because of the time it takes. You have to go through generations. And the good gene also brings in a lot of bad genes with it which you want to remove. But now you can just mutate your gene with on the basis of the knowledge that you have gained from your genomics work and modify it, and you can do it in 4 to 5 years time. Pallav, here is a test for you. You are a botany graduate. One of the petri dish contains transgenic seed, and the other one is non-transgenic. Can you, by looks, differentiate between the two?
Let me see. Both look very similar to me. Same size, same shape, same color.
They have to be, because this gene is not to increase the seed size or change any trait. It is for only one purpose, to put glyphosate resistance into the plant.
I would say this is the GM seed. Am I correct?
Let me check. There is a dot on the transgenic ones. This doesn't have any dot on it.
Okay.
This has a dot on it. This is the transgenic.
Oh, this is transgenic. So I failed the test.
No.
Which also means that for most people, on the face of it, they don't look different.
No, they should not. Unless you have bred them for a change in their shape or size, you know. But here we are just putting a trait which should not change anything else in the plant except give it resistance to glyphosate.
And that is what in your lab, you wanted to do.
Well, that is the idea, to tackle Gauravbenke problem, which is affecting almost 1.5 million hectares in Rajasthan, in Jhansi area, and in rain-fed crop is under tremendous stress from this weed.
So having failed this test is not a bad thing for me. I could not make out the difference between a genetically modified mustard or genetically engineered mustard and a normal mustard. Having understood what exactly is the work which Professor Pental has done on genetic engineering, let's go into the full-fledged growth chambers. This was a tissue culture facility into the growth chambers to see how mustard or brassica is being grown and in off-season. We are inside a special chamber where brassica or mustard is being grown. What is special about this place, and why do you have to grow it in these artificial conditions, Prof. Pental?
Pallav, we have mapped many important traits in exotic germplasm, and these transfers are being done by sexual process, what is generally known as conventional breeding. But the idea is, when you want to transfer a character, it requires about 6 generations. If we take it only in the field, 6 years is a bit too long. So to shorten that, we grow these plants under artificial light, and we get at least 1 generation during summer. And now, of course, there are lighting chambers available where you can get rapid generations, and 3-4 generations you can complete in a year. So that will help us in breeding a disease resistance gene into our varieties, bringing in quality traits into our. So this is going to be very useful technique for the so-called speed breeding, which can be done in the chambers to shorten the period of developing new varieties with interesting traits.
Like in India, mostly we see mustard being grown in winter, meaning you can grow it also in summer using these chambers?
Yes. We used to go to Leh actually earlier, but now to transport so many people and to look at the germplasm carefully and see that the plants are, because one cannot stay there for the whole period of time. So these chambers are very useful, and this is what we call as back cross breeding and generation advance, quick generation advance.
Now, you told me there is no genetically modified mustard here, but did the work on genetically engineered mustard happen here in this?
Yeah, you bring it in the confinement and take all the precautions till it is allowed to be taken out by the statutory bodies. So these chambers are very good, because when you're doing genetic transformation experiments, you cannot tune it to the season. You're doing year round, and anytime your transgenic comes, you put it in a pot and put it in a chamber. It's isolated, and it produces some seed which you can then handle whatever way you want to later on.
One of the concerns which has been around genetic engineering has been the issue of patenting of genes and patenting. Do you think there has been overzealous patenting and that has led to some backlash?
You know, actually, I find it very tragic. Rather than GM or no GM, we should be more concerned about the traits, how to acquire them, how to get the proprietary germplasm, how to introduce into our varieties. What you are saying is a very important issue, because as I mentioned to you earlier also, each crop requires a number of traits.
Correct.
And if each trait has number of patents on it, you will keep on fighting only the patent battles. That is really the major issue. And how much public funding and how much private funding. These are the two issues. The third issue is it should not be toxic or allergenic. Rest issues are all blown up and motivated by some other things rather than biosafety actually.
But this patenting issue, if like the mustard you have developed, is it protected or is it open source, or how is your genetic engineering product?
It was protected, but the Barnes Barstar is now out of protection. Glyphosate resistance is out of protection because the time period, transgenics are being grown since 1996. It's not a yesterday's story. The point is that these technologies, you require more, and if they are too much under patent regimes, there is a question mark on how to utilize them to the best of benefit to humanity. But I feel the way to do that was through CG institutions, like we brought the...
Which is the CGIAR institution.
Yes, wheat and rice revolution. Same way we should try to bring a revolution through these technologies, through international effort and through a collaborative effort which involves the whole humanity.
Now, this is non-GM. Is genetically modified or genetically engineered mustard safe?
I had put out all the documents for you to see also. There have been such extensive studies done on the Barnes Barstar system. Plus, it is already in the field in about 12 million hectares every year. Since 1996, of course, the acreage has gradually increased. But how can this be unsafe when there is so much knowledge and information about it, and our own scientists, our own GAC, RCGM have looked at it so carefully, in fact over carefully.
India has one success story on genetically engineered plants, which is cotton, BtCotton. But the work on BtCotton continues. Let's go and see how better varieties of cotton using genetic engineering are being developed. About 2 decades ago, India introduced genetically engineered cotton. Behind me are cotton plants which are genetically engineered, but where research and development is going on. What is happening in these plants, sir?
We want to have increased dosage of cry-1AC protein so that the insect is not able to break the barrier. See, the insect pests are notorious. They develop resistance very fast. And so far, BtCotton has stood the test. But for pink bollworm, for example, resistance is not up to the mark. In these plants, which is in an Indian variety, the dosage of cry-1AC remains very high till the boll formation occurs. And more expression is in the leaves and boll rind, not in the consumable parts.
Most recently, we have seen golden rice, which is genetically engineered rice, being introduced in Philippines.
You see, there, that's a good development. A lot of interesting scientific work went into that, and it will improve the nutritional status of people who have vitamin A deficiency. But I am more concerned, if you ask me, about tackling the pests of the crops so that we don't lose yields.
We also saw, while India put a moratorium on Bt Brinjal, is it correct that our neighbor Bangladesh has permitted genetically modified brinjal or eggplant to be grown in farmers' fields?
All of it has cleared all the biosafety issues. So the choice is either you have Bt Brinjal or you have pesticide laden brinjal. So, and the farmers would love to grow it. You know, if it is released in India, all farmers will like to grow brinjal which is protected by genetic means and not by chemical means.
Your own work on mustard, which is such an important oil seed crop, do you think India should embrace and release genetically engineered mustard into farmers' fields?
Absolutely. I have no doubt about that. The GAC said it in very clear terms in 2018, around 17, 18 area, that it has met all the biosafety criteria. We spent so much money and the public money on doing so intensive biosafety analysis on the mustard. And if India wants to increase productivity, hybrids is the way to go. And the best way to make hybrids in mustard is through the Barnes Barstar system, which we have already ready.
Correct.
And it will certainly increase the yield by around 30 to 40% in a very short period of time, because farmers will take the technology. See, it is public sector funded, so the companies can compete for providing seed to the farmer. And I told you, in the diversity side, we have created huge amount of diverse lines. So there are combiners available. If a good pollination control mechanism is available, it will be like in Canada, where all hybrids are, all rapeseed is hybrids now. Same will be in India, and the farmers will be very happy. That I can assure you.
Thanks a lot for speaking to us, Prof Pental,
Pleasure.
and for explaining to us the way science of genetic engineering works and the way India should progress using that.
And
Thanks a lot,
benefit
sir.
from that. Yeah, great pleasure talking to you.
My pleasure. India is already over a 1.3 billion population country. In times to come, the population is likely to go above 1.5 billion. Feeding such a large country is not easy, and all methods necessary for increasing the yield need to be deployed. And genetic engineering, gene editing are proven scientific technologies which can be used for India's development. Low input, high output agriculture is what is the need of the day. Keep watching India Science.